Okay, so this is lesson 8, genetic mutation. So when it comes to talking about genetic mutation, we usually refers to the changes that are being made to the genetic material. For example, maybe one of the nitrogenous bases being changed. And when we categorize mutations, there's so many different ways or aspects that you can look at it and you can classify it into different categories. For example, we could classify mutation in terms of the effect. So then it will be falling into 3 major categories. It's a beneficial mutation, or it's a neutral mutation, or it can be a deleterious mutation. So that looks at the effect of mutation alone. However, if you want to look at the mutation in terms of where the mutation is actually happening, or how big the scale is, you can categorize it into gene mutation or chromosomal mutation. So gene mutation is a mutation that arises from one single nitrogenous base inside a gene. And chromosomal mutation or chromosome mutation are mutations that actually changes the structure or the number of chromosome. It's much bigger scale. In gene mutation, we can, when we look at a point mutation where a single change has happened, that's starting from a gene, there are 4 different mechanisms of doing it. So we can categorize it again in terms of how it actually happens. So one way is substitution, the other way is insertion, and then you have deletion. I'm going to let you read this on your own. I'll just explain the differences briefly. Substitution, when you only change one amino basis, you actually have a relatively small effect on the other codons. So the changes usually happen to one single codon, and the effect doesn't really carry on that much. Insertion or deletion, when you're adding in one base or deleting a base, you produce a frame shift effect. So not only is that codon changed, all the rest that follows down the stream is also affected. All right, so this is a frame shift effect, important word. In terms of point mutation, you can also classify it in terms of outcome. All right, so we talked about classifying in terms of the mechanism. Now we focus on the outcome itself. So if you don't have mutation, this is a normal codon and then a normal amino acid. Now, if you look at the outcome, a point mutation where you're substituting a base or adding or deleting a base, you can either produce a silent mutation, which is, yes, the base has been changed, the codon has been changed, but if you look at the codon table, remember there's several codons coding for the same amino acid. So in this case, although it's been changed, it's still coding for the same amino acid, lysine. So we call this a silent mutation. The overall protein that are being produced by this gene will not change. It can also be a nonsense mutation. That's a very bad one, because nonsense means that you have changed the codon in a way that instead of producing amino acid, you are coding for a stop codon in translation. So gene expression, remember the last topic we did? It's going to stop here because you're coding for a stop codon. That means a release factor will come in and stop that gene production, gene expression. A missense is when your codon has been changed in a way that codes for a different amino acid. For example, it could be coding for an arginine or something else. Conservative, non-conservative, you don't really have to know it at this point, but it means that it codes for a amino acid with a different polarity, if you remember what that means from the beginning of the lesson in Unit 2. Okay, after talking about mutation that initiated or started from a gene, we're going to move on to chromosome mutation. There are two different types. You either have numerical or you have structural. So let's talk about numerical first because this is the one that we've come across before. Numerical mutation in chromosome means that an individual will have a different number of chromosomes. So instead of having a normal pair, they may have only one or they may have three of the same homologous chromosome. We had an example before, if you remember, Down syndrome is a trisomy. So that means it has triple the amount of a particular chromosome. Chromosome number 21, instead of having two, just like a normal human being, the individual has three number 21 chromosome. So that is numerical, pretty simple. And you also have structural. Now, structural mutation in chromosomes, there are different ways that it can happen. You either delete a whole section of a chromosome, just like this one right here, or you insert a whole section. This can be a duplication of a particular section. And then you can have inversion, where you swap the order of two sections in a chromosome. Or you can have a translocation. So for example, a section of this chromosome somehow is moved to a different chromosome. So these are the four different types of chromosome mutation. And that's about it in terms of the different categories or how to categorize mutation. Remember, we always look at it from different point of view. So if I ask you, hey, can you classify this mutation in terms of its effect? Then you can tell me, okay, this is a beneficial mutation or this is a deleterious mutation, which causes harm to an individual. If I ask you to define it in terms of the mechanism of how this happens, then you'll tell me, hey, this is a gene mutation or this is a chromosomal mutation. If I say, hey, if this is a gene mutation, what is the effect? Then you can tell me it is a silent point mutation, which means it doesn't really affect much, all right? So after you've done that in your notes, I want you to look at this example. This is sickle cell anemia. There's a video that talks about a patient living with sickle cell anemia. And I want you to figure out what the normal sequence, DNA sequence look like on the template strand. What is the mRNA like, so the codons, and then what is the amino acid that is being affected by it. And then I want you to look at the mutation in terms of the mechanism and the outcome, right?